Students of B.Sc 3, Semester 5, Paper 10 CBCS syllabus is Microbiology, Plant Pathology and Mushroom Culture Technology. In this unit number 4 is Mushroom Technology. In this unit number 4, today we will see about cultivation technology, storage and nutritional value of the mushrooms. We will start to learn this topic from Cultivation Practices for Pleurotus sajar kaju. It is grown indoors in available room, garage or shed with adequate light and proper ventilation. The cultivation practices involves some important steps. First step is spawn production. The propagating material used by mushroom growers for planting beds is called spawn. The spawn is mycelium of selected strain. It can be obtained from a recognized laboratory or research institute. For Pleurotus sajar kaju, mushroom spawn is prepared on wheat or jawar grains. Grains are soaked in water overnight and boiled them to become soft. The soft grains are spread over cheesecloth and 2% calcium carbonate is mixed in these soft grains. These grains are filled in empty milk bottles or heat resistant polypropylene bags. They are properly plugged with cotton plug and opening is covered with plug of paper piece tied in rubber band. These bags are sterilized in autoclave at 121 degrees centigrade. Spawn obtained from laboratory is inoculated under aseptic conditions. The bags or bottles are incubated at 25 degrees centigrade temperature. The mycelium penetrates the grains within 10 to 12 days to produce proper spawn mass. It should be used for inoculation. Observe this figure. Pleurotus sajar kaju mushroom, dingri crop in flush, spawn of mushroom. Pleurotus sajar kaju is also called as a dingri mushroom. Second step in mushroom cultivation is substrate or compost. The substrate on which mushroom grow is called compost. It includes cereal, straw, corn cobs, bagasse, banana leaves, leaf litter, waste paper, cotton waste. Paddy straw chopped into smaller pieces is most commonly used material for this mushroom. So paddy straw are used as a substrate in mushroom cultivation. It is soaked in boiling water for 2 hours, gives best results. In this compost, organic and inorganic fertilizers are also added. All the ingredients of compost are mixed well on a cement platform to ensure good quality mushroom compost. Composting may be done in open space or inside a room. Compost is prepared by long-term or short-term method to prepare well decomposed heap. This is done by turning with frequent watering. This slide shows standard formula for compost. Substratum or compost for mushroom cultivation includes wheat straw or rice straw 300 kg, calcium ammonium nitrate or ammonium sulfate 9 kg, urea 3.6 kg, potash 3 kg, superphosphate 3 kg, wheat bran 30 kg, gypsum 30 kg, Firadon 3G 40 ml, BHC 5% dust 250 gram and molasses 5 kg. The third step is growing site. Pleurotus species are usually grown in indoors and any ventilated room is used as a growing site. In humid climate, the house made up of bamboo or mud plaster are recommended. The floor of such house should be of cement. The roof is made up of the thatch layer. Room must be with ventilation. Inside the room, shells are provided with 3 tiers with sufficient space from walls. The fourth step is pasteurization. The compost is pasteurized at 45 degrees centigrade for 24 hours. Trays are filled uniformly. While filling trays with compost, spawn is mixed layer by layer. The temperature is maintained at 22 to 28 degrees centigrade. Mycelium starts growing within 6 to 7 days and the trays are ready for casing. Next step is spawning. The spawn obtained from laboratory or produced is mixed with substrate or compost 2 to 5% of the wet substrate. The spawn spreading should be done carefully under aseptic conditions. Sixth step in mushroom cultivation is filling. We can cultivate mushroom in tray or polythene bags. The trays and bags should be perforated by cutting holes to permit ventilation within substrate. The bags or trays are stacked in shelves. During this process, care should be taken that temperature inside should not exceed 30 degree centigrade. Then the seventh step is the crop. The packed bags or blocks are incubated in ventilated room at 25 degree centigrade temperature and 80 to 85% humidity. The mycelium penetrates the substrate completely within 12 to 14 days. The covering is removed and blocks are arranged on shelves. The humidity is maintained by sprinkling water on floor or walls. The spawn or blocks of substrate can be covered with gunny cloth. Light is essential for the fruiting. Smaller pinheads will appear within 2 weeks and there is a development of mushroom within 20 to 25 days. The next step is harvesting. The harvesting is done when cap is fold having diameter of 8 to 10 centimeter. The base of tip is cut by using knife. Now the mushrooms are ready to sell in market. We can cultivate Pleurotus sajar kaju mushroom by using limited resources and can earn good money. This slide shows information about advantages of growing Pleurotus sajar kaju. It can be grown on moist of the cellulose farm waste. Softening of substrate is sufficient for cultivation of mushroom. Casing is not required. Variety of materials are used for the cultivation. For example, polythene bags, nylon net, trays, baskets, shells. Spawn runs fast and crop can be harvested within 2 weeks or 20 to 25 days. We can obtain higher yield within wide range of temperature 22 to 28 degrees centigrade. Mushrooms are very useful. Some value added products are present in mushroom. Pleurotus or oyster mushroom are rich in proteins. It is with minerals like calcium, potassium, phosphorus, sodium. It serves as a good source of iron, folic acid, vitamin B, C, D, K, riboflavin, biotin and thiamine. Dried forms of mushroom are powdered and used for mushroom soup preparation. This soup increases appetite. Frozen mushrooms are used in a variety of foodstuff articles. Mushrooms like Pleurotus can break down and make it upgraded quality feed for animals. Some delicious crunchy mushrooms biscuit are prepared using some other ingredients like maida, sugar, ghee, baking soda, milk powder and ammonium bicarbonates. Mushroom ketchup and mushroom candy are also prepared by using fresh mushrooms. Mushroom preserve, muramba, mushroom pickles, mushroom papads are prepared by using standardized formulations of Tamil Nadu Agricultural University, Coimbatore, Tamil Nadu. Snacks, finger balls, mouth-watering products are prepared from fresh and dried mushrooms. In this slide, we will study about commercial importance of mushroom. Mushrooms are very important because it is used in various fields. Food value. All edible mushroom species are rich source of protein, mineral, vitamin and carbohydrates. Its medicinal use. Mushrooms show antiviral, antibacterial, antifungal properties. Hence, they are used as a medicine to treat eye diseases. Species like Flammulina volutives are used to reduce growth of tumor cells and to control high blood pressure. Mushroom is the best source of dyes. Polyporous sulfurous yields yellow color of dye. While Fomes igniarius is used to prepare blackish brown colored dye. So mushrooms are used as a source of dyes. Then its ornamental value. Many mushrooms have fabulous shapes, size and colors. These are used as a decorative materials. Mushrooms increase soil fertility. In nature, it brings about decomposition of dead matter and humus content of the soil get increased. Soil becomes more fertile due to presence of the mushrooms. Recycle of nutrients is one important role of mushroom. In nature, mushrooms brings about decomposition of dead matter. Recycling of the nutrients takes place. Then it provides job potential. Mushrooms are sold at high price. So students, unemployed youth and retired persons can take mushroom cultivation as source of earning. It will be a good industry to supply nutritious food. So you know that it is rich in protein, mineral, vitamin, carbohydrate. So it is very useful as a nutritious food. Thank you.